Okay, so we're in there. All right. So again, that's the sequence you go through, right? You guys, you can take pictures, right? So you have adaptive radiation, you spread outward, you become geographically isolated. Geographic isolation results in selective pressures, you become genetically different, and now you've allopatrically speciated, right? That results in reproductive isolation. And that you're officially two new species when you can no longer successfully interbreed, okay? So I thought about this visually, and so I quick put together this little map of Europe, and I made up a fictitious population of birds or whatever, right? That started off right here in Central Europe, right? And what happens is, again, they radiate outward, right? So now there's a population A that goes up to Britain, right? They want some fish and chips. There's another one that goes over to Spain. There's another one that goes here to North Africa where it's nice and warm, right? Another one goes to Italy where it's great food, right? And then this last population's over here by the Black Sea or the Baltic, right, area, and it, resides over there. These 5 populations are now separated from each other, right, and they're going to adapt, and they're going to become genetically different. So first they go through geographic isolation, which leads to allopatric speciation, right? Species A and B may not be able to ever interbreed again because they're experiencing such different environmental pressures that there are different alleles now that are being selected for. So there may be a different group of alleles here than what you find here. And then these guys are the same thing, right? And they're geographically isolated, right? There are definitely mountains here. You got the Swiss Alps. I don't know, there's something else over here. You got this big body of water, and then just distance, And so eventually they experience such different, dramatically different environments that they become completely isolated from each other. Not only geographically, but also reproductively, okay? This is the same thing that happened with Darwin's finches, right? The finches started on the mainland, and they all radiated to different islands where they were geographically isolated. And then they speciated allopatrically because they were experiencing unique conditions in different places, okay? Are we all right with that? We're good? Okay. You want to take a picture, or you got it? You good? Okay. Oh, go ahead. Okay. And so all of this is happening because you've got changes in your population. Your population is migrating, right? So we have gene flow, right? Genes are flowing out of here, right? We're going to new places where there's selection, right? So now some alleles that once had no impact now are selected for, right? It's an advantage. And maybe other alleles are not so much of an advantage. So these populations, their frequencies of alleles, the percentages of alleles are going to change what you originally had, right? And then maybe in these new locations, maybe there are different types of selective breeding going on, right? Maybe different characteristics are now more beneficial, right? And so we're selecting those as well. And again, these moved out from a big population, and now they're smaller, right? So there might be some changes going on there, right? Small populations, what does that lead to? If your population is too small, what could happen? Right, you can have genetic drift, right? You can just kind of like, by chance, randomly lose alleles, right? And that becomes a major issue, right? So we don't want that happening, right? But it does occur in a small population. Genetic drift, there are 2 things that can happen: your founder effect and your bottleneck effect, right? Can anyone tell me what the founder effect is? We'll give you an example. Go ahead.
When a bunch of organisms live in a population somewhere, and all of them have a certain set alleles that is representative.
Right. Yep, exactly. So it's when you have a population that fragments off and goes to a new location, and maybe this is a very small group, and it doesn't represent all the alleles here, right? It's less diverse, okay? And so now this group here, they're the founders of a new colony or a new population, right? But it's not the same genetically as what you had here, okay, at the initial, right? That's your founder effect. That's what happened with the Amish in Pennsylvania, right? They fragmented off from their group, which I think was like in Germany. I'm not even sure. And then they moved to the United States. They established themselves in central Pennsylvania, and they stayed isolated, right? And they were representative of just some of the alleles. And because of that, they now have limited genetic diversity. They have a high rate of achondroplastic dwarfism and a high rate of polydactyly, which is having 6 fingers, right? Yeah. So perfect. I mean, it's like a wild example of a founder effect, right? And those alleles are not representative of what you see outside, okay? And then the other one is the bottleneck effect, right? What's a population bottleneck? Well, what would be an example of that? Yeah, right, they're overhunted, and they're almost overhunted to extinction, right? And so what happens to that population? Bottlenecks down. So what's bottlenecking down? It's what? Right, the allele frequencies, right. So some of the alleles are actually bottlenecked down to 0, right? You lose alleles, right? And so it's less diverse than what you originally had, right? So endangered species have all experienced bottlenecking effects, right? Where the population's been reduced and it's not representative of what you originally had, okay? And that's dangerous because you can have a lack of diversity means that your population's unstable, right? Large population size, that's what eliminates genetic drift, right? You guys remember the 5 main parts of the Hardy-Weinberg equilibrium, right? Number 1 is no mutation, right? Because Hardy-Weinberg, right? Hardy-Weinberg says that your allele frequencies will stay the same. They won't change if you have no mutation. What else? No what?
Random mating.
You'll have random mating, right?
No large population.
Large population, good. No natural selection, right?
Is there like no migration?
Yeah, no migration, right? You don't want any alleles coming in or leaving, right? So you're keeping everything stabilized and the same, right? So you want it to be isolated, right? An isolated population, you're not going to see that happening, right? So what do we have so far? We said no mutation, no migration, large population, random mating, and natural selection, right? No natural selection. So you want all of your alleles to be equal, right? It doesn't matter if you're dominant or recessive. Every allele has equal survival chances, right? That's Hardy-Weinberg, right? So the opposite of that would be evolution, right? We have mutations, you have gene flow, you have species moving to new locations, you've got new alleles coming in, right? There is natural selection. That would cause a population to evolve, right? And the Hardy-Weinberg equation, this is what gets people a little tripped up. So I want to go over some of the terminology with you guys so that you're comfortable with it. The first thing is you have to figure out what Q is equal to. Okay, always solve for Q. You can figure out Q by doing the square root of Q squared. Now, what does Q squared represent? It's a recessive phenotype, right? It's also the recessive genotype. So if they give you a situation where they say you have plants that have purple and white flowers, first they have to tell you which is dominant, right? And they'll tell you, white is recessive. Okay, and then they'll say that in this population we have 1,000 plants and 340 of them are white, right? What are they giving you there? 340 out of 1,000 is Q squared, right? So they're telling you what Q squared is, right? 340 out of 1,000 is 34%, right? So Q squared is 34%. Then you figure out Q by doing the square root of that, okay? 2PQ and P squared, that's your dominant phenotype. So remember, the dominant phenotype is big A and big A little a, right? So for purple plants, it would be both the homozygous dominant and the hybrids, okay? But you need to figure out what Q is, and then you can figure out everything else. So usually you figure that out by figuring out Q squared, okay? And we're going to go over a couple of those problems. We'll do them now. So you guys have that packet that I gave you from the other day, right? If you need extras, I have them. So I have these and the answers. And what we're going to do is we're going to jump around a couple of these because there's really like 3 that I want to look at just to make sure that you guys are solid. So when you open that, and if you need extra copies, I have them up here. We're going to go to question 2. Now, you guys at home, I have set up on Schoology these questions for you guys, okay? So the Hardy-Weinberg practice problems are in week 28, which is the week we're in right now. And you can click right here, practice number 2, and open that up, okay? I have the answers also. They're not in Kami because I'm not collecting it, but I figured you can just kind of look at them and just work the problems out just like on a piece of paper on your own, okay? So let me just pull this back up so you guys can see. All right. So you have a sample of 2,400 people. 1,482 were found to have the dominant taster phenotype. So this is dealing with the ability to taste the PTC paper or not. So they tell you, assume it's in Hardy-Weinberg, how many individuals are heterozygous for this trait. So we know that tasting means that you're dominant, right? So what are they giving us here? 1,482 out of 2,400. What does that represent?
Square.
And what else? Yeah, it's P squared and 2PQ. Because remember, people that can taste are going to have two dominant alleles, or they're going to be hybrid. Okay? So remember that 1482, that represents P squared and 2PQ. Okay? The number that they give you. Well, actually, the percent that they represent in the population. Okay? So now we have to figure out what is Q squared equal to, right? So Q squared, how do we figure this out? Yeah, subtract 1482 from 2400, right? And what number do you get? Right, that's 918 people, right? But now we need a percent. Always remember that when we're dealing with frequencies, we're dealing with percentage. So when you see the word frequency, just click that into your head to percent, right? So Q squared is not equal to 918. It's 918 out of 2400, right? That's what Q squared is equal to. And that's going to get you a decimal, right? And tell me what you guys get. 0.38, right? So it's about 38%, right? 0.38, okay? So that's Q squared, right? How do we get to Q? Right, got to square root this thing, right? So the square root of 0.38 is Q. So Q is equal to I think it's 0.62, right? Right? I've done this enough, so it should be, if I don't memorize it by now, I should go home. All right. So Q is 0.62, right? What is P equal to then? Right. So P is equal to not 48, because that would take it over 1. 0.38. Yeah, so P is 0.38. Okay. So now we've got P and Q. Does anybody that doesn't get how we got these? Are we okay with it? All right. So now we have P and Q. Now we have to actually answer the question. And they want to know how many are heterozygous. So we have to figure out what 2PQ is. So we just plug these in, right? 2 times P times Q, which is 2 times 0.38 times 62. Okay, and you're going to get a number. I think it's like 0.472 or something like that, right? Okay, so that's the percent of the population, but they want the number. So how do you get the number? Not divide, multiply. Right, 2400 times 0.472. I got 11.1133. Is that what you guys got? Good? Okay. Okay, so 1,133. That's right. And that's the number. So that's choice C. Okay. Do you see how we did it? We're right with it? We're good? Okay. I'm going to go to a different one, which isn't as obvious. So here we go. Let's do number 9. Number 9's got some weird numbers to it. But I like this because it's asking you for something kind of different. So they just came up, they didn't even give you a name of the disorder. They got lazy. They're just like, yeah, there's a genetic disorder out there. We're not even going to name it, but it affects 1 in 6,600 people. And it says it's a single gene that has 2 alleles. If you're recessive, you're affected. So we know that the recessive is going to be 1 in 6,600. So now we have to get a percent out of that, right? So they're telling you that Q squared is 1 in 6,600, right? Because that's your recessive. So now you got to do some math, right? So 1 divided by 6,600. Yeah, it's like a really big, long decimal, right? It's 0.00015, right? Yeah, that's Q squared, right? And now you got to figure out Q. So how do you do that? square root, right? All right. So what do you guys get for that? Again, don't be like thrown, like one of the things they try to do is throw you off by giving you these like weird numbers. Just plug them in, work it out, it's fine.
0, 1.
What'd you get? You got it?
I got 0.012225.
I don't know. I didn't do this one. 0.01225. Yep, that's right. Yep. So 0.0123, right? Okay, that's Q. So what does P have to equal?
You're gonna see equal whatever 1 minus.
So it's got to be
0.987.
Yep, 0.9877. All right, so that's P, right? So that's a dominant allele. They want to know what is the frequency of carriers in the general population. So what defines a carrier? Go ahead.
It would be the one that has only one recessive, or like a hybrid.
You said hybrid, right?
Or hetero.
Yeah, yep, heterozygous hybrid. So what they're asking you here is who are the carriers? Those are your hybrids, right? That's 2PQ. Because remember, carriers don't have the disease, but they can pass on the recessive, okay? So this is going to be 2 times 0.9877 times 0.0123, right? And then you're going to get a crazy decimal out of that, okay? And hopefully the decimal that you get is choice C, 0.02430, right? Okay, and there you go. Do you see how we got that? Go ahead. Yeah, absolutely. Go ahead. Do you guys see how we got that? How do we feel about Hardy-Weinberg equation? You're going to have the formula on the test. So I'm giving it to you on the test. You'll see it. You have it on the AP exam too. So you don't have to memorize it. Go ahead, Lynn.
Can you go over the one on like the AP classroom? Do you know how Hardy-Weinberg one on it?
Yeah, the one that.
It was like I got 0.06, but the answer's like 0.03.
Which one was that? On the AP classroom?
Yeah.
Let me see.
It was the first one, the A one.
Yep, let me pull that up right now. Let's see. That was the progress check A?
Yeah.
Okay.
The A one.
Okay, and it was,
After.
let's see, let's do this.
How about a graph on it?
Watch, it's like the last one. Is that it?
No. I could have swore it was an A.
It's okay. We'll go to B.
I swear it was an A.
Oh, you know what? Wait, was that it? Let me look. Let's see. 2. Was it this one?
No,
No.
it had like the answers were like 0.03 and then 0.06.
Okay, let's see.
What? I swear I did that.
Okay. It's not there.
That's
Hmm.
okay. It's fine.
All right, we'll figure it out. Okay. Okay.
What?
So are we good with this? We're set? We're good? Okay, excellent. So there's another one I wanted to just do a quick recap on. Let me go to, actually I should have stayed in AP classroom. Hang on a sec.
I handed on the word, so. I did. Let's see what I did. Awesome.
Let me go to my downloads.
Yeah. Because I can get it after the explanation. But my chances of having to go. Alright, Nancy, like you can do.
Yeah, this is it. All right.
I think I can find out.
Is she gonna change her name?
Okay.
75. Like, as.
I know, but like,
How do we feel about cladograms? The phylogenetic trees. I heard an okay, we'll do that real quick.
When they're already filled out for me, otherwise.
Oh, like this one, right?
That's much.
Right. Right. So you got this table, and it shows the characters for these organisms, and they want you to indicate which is the best evolutionary relationship. So to set this up, right, you have to go to your out group, right? So the out group is the one that's has the least similarities. So without looking at this here, which one has probably the least similarities, right? So if you look at each organism, you have the plus signs of what they have. This shows you what's derived and what's present. So which one is probably going to be the out group? All the way to the right, to the left. Go ahead.
The fish?
Yeah, the fish, right? Because it's only got one thing there. It's got a jaw, right? So what would be the next thing? Probably amphibians, right? And then after that, let's see, you got 1, 2, 3, 4.
The mammal?
Crocodilian. Right, crocodilian, right? Because you've got 1, 2, 3, 4 there. So you have 1, and you have 2, and you've got 4 here, and then you've got birds and mammals left. Now the question is, do birds go here or on the outside?
There, because no one else's feathers on them. No, I don't know.
Well, look at the similarities, right? Birds have similarities with everybody here. Crocodile. Here's one here. So this is what makes birds and crocodiles different from mammals and crocodiles. See that? You see the difference? So which of these is the correct one? Is it A? Let's see, B. That's way out, right? Ah, look at that. The answer was there, A, right? That's your out group. Do you see how that works? Are we okay with it? Yes, no, what? Yep. You got it? All right. There's another one I wanted to show you. I'm just trying to find the picture. Here it is. Okay. Here's an example here. Okay, so you have these fish populations, right? There are 5 populations of fish, A, B, C, D, E, right? And the scientists are proposing that A is was the ancestral original species for all these other fish. Okay, so based upon this, what could likely count for the origin of species B in that lake? What type of evolutionary action? So I'm going to just zoom this out so you can see the choices. Okay, hopefully you guys can still see that. Okay, so A and B are in this pond, Lake 1, and here's Lake 2 with the other one. So what would most likely be the result, the origin of species B in that first lake? Do you guys see it? Okay. What do you got, JP?
I think, but I don't want to be wrong.
Say it.
Well, I was going to say allopate.
Patrick?
Yeah.
Okay.
Association.
What does allopatric mean? Allo means different, right?
Yeah.
Place. So that means they would evolve in a different place. So A and B are here, right? B's not here.
Different species.
Different place. So Patrick means place. So is that allopatric?
No.
I just said the answer. It's sympatric because they're in this lake. They're evolving from that same spot in that lake, right? So these guys, it's allopatric because they diverted from the original species and then they evolved here, right? Or they became different species here. But here, they're locked into that little lake. So A and B, they're together. B developed within that lake with A. So that's considered sympatric. It's in the same place. Does that make sense? Right? Right? Because they're locked. I like this picture because they're kind of like, they're locked in there. They can't go anywhere. So that's where they evolved. And then what happened is like there was a little stream that occurred every once in a while where A was able to get in here. And so then A made its way in here. Right? And who knows, D, C, and E may have already been present. You know, we don't know. But the fact that you have two species in that one little spot and B isn't anywhere else here means that they both evolved within the same population sympatrically. Okay? We good with that? All right. There's one more I saw that I wanted to go over with you guys. Oh, this one. Okay. Butterflies, right? So, you okay? Oh, it's killing me. I know. I was like dying this morning. And then it smells out there too. But all right. So you have these two models, right? So which of these has evolved in a situation where there were gradual changes in the environment and things progress very slowly? The one on the left, right? You're right. The one on the left, right? This represents gradualism because what you're seeing here is you have one, ancestral form, and then it diverged into these two, and then it gradually morphed into the species that you see today. Okay, this represents a situation where you had your ancestral form, and then there was rapid change, and then here nothing was happening. Okay, and in the case of this one, it stayed stable like up to present day, and then this was stable for a long time, and then it split here, and that's how you got these two forms. Okay. This is punctuated because punctuated means it's happening at points. So that evolution happened here and here. That's where you had evolutionary divergence going on. Here, it's happening at a constant rate and it's very gradual. So that represents gradualism, and that's your punctuated equilibrium. Okay with that? Is that good? All right. And then there was another one that I had. So we're okay with that. Is this? Oh. Oh, this one. Okay. So you see a graph like this, right? Your initial population was this dotted line, and then it narrowed in a little bit, right? So this is favoring a phenotype that's in the middle. Remember, you've got three different things here. You've got one extreme here, the middle, and then the other extreme. So what kind of selection does that represent? Where you're really allowing that middle phenotype to really pop and really survive. And you're putting pressure on these two. What kind of selection is that? Is it stabilizing? Or is it disruptive? Or is it the heterozygote advantage? Those are your three choices. So are we stabilizing here? Are we having directional selection, or are we doing heterozygote advantage? Yeah.
I just want like heterozygous because it's in the middle.
Yep, that's it. It's heterozygous because it's in the middle, right? When you take this bell curve and you tighten it up like this is telling you that the hybrids in the middle have the advantage, and that you're selecting against either extreme, either little a, or big A, little a. Okay, so that's stabilizing selection. I'm sorry, that's the hybrid. Stabilizing selection, which is hybrid vigor or heterozygous advantage. Okay, if it's shifted one way or the other, that would be considered directional. Okay, where you're favoring one end versus the other. That's directional selection. Okay, because you're going one direction towards the other. Right. There's actually a picture of that up here that I saw, which I'll just double back to. This one. It's where they took these wolves or dogs, or I don't know what it was, something. There we go. Foxes, right? They took foxes, and the wild population is very aggressive, And so what they did is they took these foxes and they domesticated them in the zoo for a while, and then they became much more docile, right? And so what we're seeing here is a change in their behavior pattern. This is a directional shift, right? It started out here more aggressive, and then they became more docile, okay? And so it shifted directionally this way, okay? So when you see this where your original population looks like this, and then you shift one way or the other, you're going either to the right or to the left, that's directional selection, okay? We go with that? All right, that's it. Oh, question?
What would the stabilizing look like?
Stabilizing is it's the bell curve that I just showed you
Oh, I
before,
thought that was the heterozygote.
which is stabilizing, yeah.
Oh, so it's the same thing.
Yeah, disruptive, what would happen in disruptive is you would see two bumps like this and nothing in the middle. So you would see both extremes and nothing in the middle. So originally the dotted line would be like this, and then what would happen is you'd have one here and one here. So that's, for an example, like with bird beaks, right? If you have two kinds of food, large and small seeds, you're only going to have large and small beaks. Medium-sized beaks don't work well,
Oh, okay.
right? Because medium size is not big enough for the big seeds, it's not small enough for the small seeds. So that gets eliminated. And that's known as bimodal or disruptive selection, because you're only seeing the two extremes being selected for. It's rare to see that, because usually hybrids do really well. Most of the time you've got a bell curve and your hybrids are selected for. And then the other thing is extremes of genetic diseases, that's it. All right, are we okay? We feel good?